Our nervous system works based on a network of wires called neurons. There are two kinds of neurons, namely axons and dendrites. Axons take information away from the cell body, and dendrites bring information to the cell body. Information from one neuron flows to another neuron across a synapse. The synapse contains a small gap separating neurons. The synapse consists of, firstly, a presynaptic ending that contains neurotransmitters, mitochondria, and other cell organelles. Secondly, a postsynaptic ending that contains receptor sites for neurotransmitters. And lastly, a synaptic cleft or space between the presynaptic and postsynaptic endings. Let us now zoom in and see what actually happens when signals are transmitted across the synapse. At the end of the presynaptic neuron, there are voltage-gated calcium channels. When an action potential reaches the synapse, these channels open, causing calcium ions to flow into the cell. These calcium ions cause the synaptic vesicles to fuse with the cell membrane, releasing their contents, the neurotransmitter chemicals, by exocytosis. The neurotransmitter then diffuse across the synaptic cleft. In the postsynaptic neuron, there are voltage-gated sodium channels instead of the calcium voltage-gated ones. So, the neurotransmitter binds to the neuroreceptors in the postsynaptic membrane, causing the channels to open. As a result, sodium ions flow in from the voltage-gated sodium channels. As a result, this causes a depolarization of the postsynaptic cell membrane, which may initiate an action potential if the threshold is reached. The neurotransmitter is broken down by a specific enzyme in the synaptic cleft. For example, the enzyme acetylcholinesterase breaks down the neurotransmitter acetylcholine. The breakdown products are absorbed by the presynaptic neuron by endocytosis, and it is used to re-synthesize more neurotransmitters, using energy from the mitochondria. This stops the synapse being permanently on.